Yeah, hi everybody. My name's Tim Rudge. I'm assistant professor in Universidad Católica de Chile in Chile. And so this workshop, as you know, is about Flapjack, which is our data management and analysis tool for genetic circuit characterization. So what I'm going to do, I guess, while we wait also for people, is just give an introduction to the project. What was our motivation for doing it, what are the goals of the project, and a bit of an introduction about what the system is. Before then, Carlos, Gonzalo, and Guillermo are going to talk in more detail about the different aspects and get you guys to actually do some stuff with the system. So we're interested in the design, build, test, learn cycle, as you know, is the kind of standard approach and a goal of synthetic biology is to develop tools and methods for each of these phases of the engineering cycle to make it as efficient as possible and as effective as possible. For example, using automation and different types of software tools to join up these different phases of the design cycle. So our motivation for building this system was really looking at this link between the test and the learn phases of the design cycle. So what are these two stages? So in the test stage, we do a bunch of experiments to measure our circuit, and this generates large volumes of data, often with different formats from different types of machines, different experimental setups and different conditions, etc. And the learn stage, the idea of the learn stage is to take that data to analyze it, to extract meaningful information about the circuit and how it operates. And that's what principally is characterization or parameterization of models in order to inform new designs and go around the design cycle. And at the moment, there's not really good standards for this interface between the test and the learn cycle. So typically, what we've seen in our lab, at least, is that people have, well, the machines and the way that we store data is fairly ad hoc, for example, in Excel sheets or CSV files, with no kind of common format or standard between them. And also, what we found is that different people, different students and different postdocs and whatever, had different scripts that they used to analyze their data in slightly different ways. So even extracting kind of common features of genetic circuits, like the gene expression rates of the different genes, people do in different ways. And so it makes it difficult to compare between them. Also, there are different tools used to do it. And so compatibility is kind of an issue. So going back to the design, build, test, learn cycle, we already have a bunch of tools for these different phases. So for example, in the design phase, we have tools like SBOL Canvas, where we can design compositions of parts to generate genetic circuits. And then we can store them in platforms like SynBioHub, which allows us to analyze the compositions, components and modules of our genetic circuit. Then something that we're doing and is becoming more common, I think, especially, I don't know if there's much discussion in the biofoundries about different hardwares to do the actual build phase and also the test phase of measurements of these genetic circuits. We're using Opentrons. There are different types of automation systems, essentially liquid handling robots, to do these kinds of tasks, to make this interface between build and test more efficient. Then before we get to the design stage of actually putting together compositions of parts, what we need to really make this cycle efficient is design abstractions, which are higher level descriptions of genetic circuits, higher level than individual parts. So for example, Cello uses the abstraction of logic gates. So a logic gate is a functional composition of parts which encodes a certain type of behavior that we're interested in our circuit. And we can, for example, using Cello, automatically design compositions of parts to encode those logics. You might have seen Gonzalo's talk this morning about his system Loica, which is another design abstraction based on this concept of operators and regulators. If you're interested, I guess you can watch the recording of Gonzalo's talk. But what we identified was really there's not much happening here in the test and learn phases, where we take this data from automated systems which can produce lots of data, and then we'd like to feed that in to parameterize these abstract elements of these genetic circuit designs. So that's where our system Flapjack 6 in this design, build, test, learn cycle is taking this data, organizing and managing it, and then analyzing it to spit out kind of useful information for this design abstraction phase, and then going around the design, build, test, learn cycle. And we would like to tie all this together with some kind of standard, and the kind of go-to standard for synthetic biology, for open synthetic biology, because we're interested in making this open and accessible, is SBOL. So what the aims of the project are is to bridge this gap between the test and learn phases of the cycle. And what we'd like to have is centralized data storage and management to allow people to share and exchange information about their genetic circuits. We would like to link that experimental data to metadata. And the obvious one is about the composition of the circuit, but also the experimental conditions. And then we would like to be able to query and kind of slice that data in different ways to pull out information about the behavior of genetic circuits. We would like to have standardized analysis methods so that, for example, if I calculate the gene expression rate and I share my data with someone, they do the same calculation, we get the same results between us. And so our design process can work in parallel with the same data. We like it to be accessible and with a user-friendly interface for all kinds of levels of ability in terms of technical skills like programming. We'd like to have a user interface where essentially there's no programming involved, but we can do the same kinds of analysis and data management that we can with more powerful and extensible programming interfaces. So having an API which can connect to other tools, for example, directly. Essentially, I guess one goal of this design, build, test, learn cycle for SynBio is to try and remove as much manual intervention as possible. So connecting those tools directly is a big goal. Then the way that we'd like to connect them together is with common exchange standards and platforms. So SBOL and SynBioHub, where we can describe the genetic circuit and store it in a way that it can be, the information about the design can be queried and analyzed, and then we can refer it back to the experimental data about that design. So just to introduce a bit briefly about what Flapjack is, essentially the basis of Flapjack is a data model to describe the test data that we get from the test phase. And so the high level object is the study, which is kind of the project or the kind of group of experiments which we describe as an assay. So for example, for a certain purpose, I might do a bunch of different assays and they would all be combined in a study. So an assay is a particular measurement process which produces measurements, and the object which you're measuring is a sample. And so a sample could be, for example, if we have a plague reader assay, a sample could be a well in the plague reader, or it could be a culture in a flow cytometry experiment, or a single cell in a single cell experiment, like a microfluidic experiment, for example. So those samples are subject to measurement, and they produce these elementary objects, which are measurements, which are single time point measurements of a particular signal at a particular time. So a signal could be a fluorescence channel, for example. And then that measurement and the sample is with respect to a particular vector, which we call a vector, which is really the DNA that encodes the circuit. And so it could be a collection of plasmids, for example, or a single plasmid, which encodes the elements of the genetic circuit. Then we need to link that to metadata about what the experiment was. So what strain or chassis organism was this? If there is any, it could be a cell-free experiment, for example. What media or substrate composition was used to grow the cells, if there were cells. If there were no cells, what substrate was used in the cell-free assay to drive the circuit, essentially. And then we have supplementary chemicals, which could be like inducer chemicals, which affect the operation of the circuit by changing the gene expression levels. So that's the essential structure of the data model. And then the architecture of the system on the right here, you can see we have on the server web app, which is basically a way to organize the data model, which is backed up with Django and a Postgres database, which provides an API via HTTP REST API and WebSockets for longer computations. And that can be accessed via our front end, the web front end. So this is a user interface, which you can see on the left, which allows querying, filtering, searching data, and then interactively plotting it and analyzing it. So we can apply different analysis methods and look at the results and interact with the results, download the graphs of the results, which you'll see directly in a way that you could put them into a paper, for example. But also this API allows other tools to connect directly. So for example, you might have a tool for the learn phase, which is doing simulations of genetic circuits, which can pull out automatically data from Flapjack. And we have a Python package which enables that. And Loika, which Gonzalo described, uses that Python package to link models to data and to do characterization of different components of genetic circuits. So essentially that's the background to Flapjack and a little bit about the structure of how it works. So now, well, I'll just introduce the team quickly who are going to do this workshop and have done the development. So Guillermo's really the principal developer of the software and the architecture. Gonzalo has principally been working with testing the system and trying to break it for us so that we can fix it. And Carlos has been doing great work with documentation to allow people to use it effectively, as well as Gabrielle, who's been doing a lot of work with the documentation for the API. Just to mention who I am, and you can contact me on this email at the bottom, and we have a web page and social media. So if you want to send us messages via that route, you can. And just to mention that we're in Chile at the moment, but next month, well, at least I will be, and in the next few months, the whole lab will be moving to Newcastle University, where we will be actively recruiting students and postdocs. So if anyone is interested, please drop me a line. We have a great master's program in synthetic biology and PhD programs focused on synthetic biology from a computational point of view. But also we have wet labs and experimental work that we're doing to kind of back up these design tools. So I'll hand over to Carlos, who's going to start the workshop with a description of the front-end interface and how to work with that. So stop sharing.
Okay. Thank you, Tim, for the introduction. So are you able to see my screen? Great, I'm here on Flapjack. So now let's review Flapjack's web interface. In this part of the tutorial, we will take a walkthrough of the main interface, learn how to format your data and then upload it to the platform, how to manage your studies, and finally, how to visualize the data in order to perform the analysis that seem relevant to you in a quick and easy way. When landing on Flapjack, you are greeted with a message inviting you to create an account or to sign in to your existing account. This process is pretty straightforward, and it will only ask you for a username, email, and password. You can do that process now if you want to follow along. For that, just go to our public instance that is available at flapjack.raj-lab.org. Since I have already created my account, I'm just going to sign in. Once you have logged in, you can access the browse page where you'll see all the studies that have been shared with you or that are public. In this page, you can search by studies or any of the other models that Tim has previously stated: essays, vectors, media, training signals. And for that, you just need to use the search box and type what you want to search. It's easy as that, and it works on all these fields here and for all the models. And finally, we have the view page, where you can view and analyze the data of the studies that you're interested in. So you have performed a study, right? So the next logical step for you is to upload it in order to see it. For doing that, we might ask you to download the zip file that we have shared on the Slack channel, because we are going to use that one. For that part, go over to your username, and this drop-down menu is going to appear where you can click Upload. Okay, but first, we have to do a little adjustment to the Excel file that you get from the plate reader. I don't know if you have the Excel file already open, or if you're going to follow along. If you need more time, let me know. So, Flapjack allows you to upload the file directly obtained from the plate reader, but you have, as I said, we have to make a little adjustments to the Excel file. As you can see, the Excel file just contains one sheet, and this sheet we must rename it to data, so that Flapjack can understand what it's receiving. Here we have the other file that it's already completed for you. So as you can see, we have renamed the name of the sheet to data from the previous name that was plate 1. Once we have already done that, we have to create 4 more sheets called media, strains, DNA, and chemicals. And in these sheets, we're going to indicate the information for each individual well with the following pattern. In the first cell, we are going to repeat the title of the sheet previously indicated, for this case, media, for an easy identification. In the other cells from row 1, we number them from 1 to 12, while the other cells in column A, we have to name them from A to H. Then we fill each cell with the corresponding information according to what was placed in the 96 well plate. If you need to leave a field without information, you can do so by just typing none. Keep in mind that if you type none on media, Flapjack is going to interpret that there's no data associated to that plate, to that pod, and the plate reader, so it's going to discard. I mean, this same procedure is done on all the other sheets on strings. As you can see here, we also have type none here because in this case it was a control. And you can also insert information from more than one plate under the same field, as we can see here in the case of DNA. And to do so, we just need to leave a blank row separating both. And as I showed you previously, as I told you previously, since no vector was incorporated in these wells, we just type none on all of this. Seeing. Okay, I'm seeing now in the chat, they already give you the link to the zip file. Okay. After you have done that, you get the Excel file that it's ready to upload. In that case, if you want, you can just go ahead and upload following us. You can select a study that has been already created previously, or you can create a new one. In this case, we're going to create a new one. You have to provide a name, IWBDA workshop, and a description. You can also provide a DOI. This is not a requirement, but it's a good idea if you have already published a paper, for example, and then you can leave the information there for the people to view it. Also, you can click here if you want the study to be public so anyone can see it. Then we have to select the type of machine that we were using. In this case, we use an HDX Synergy. And finally, we have to select the data file that we're going to upload. In this case, I'm going to go ahead. I think I need to close this first, because otherwise we might have a trouble. To select the edit file with all the sheets that we have previously created. Now it's going to ask us for the assay. In this case, you can put a name for the assay for USI identification and a description. A more descriptive description will be more useful, but for the sakes of the tutorial, we're just going to leave it there. I'll leave it as that. And for the temperature, you have to indicate the temperature at where the experiments were performed. Then we click submit. In this case, now Flapjack has recognized the inserted fields, and we can choose their values from the drop-down menu. But if a value, and then we can search for the value for easy identification using the search box. In the case that a value is not present, we can create it by using the button Create New. In this case, we have to provide the name, and also you can also provide an ASP for URI. I'm going to go ahead and complete this. Now we have to do the same for the chemicals. As I stated before, if a chemical is not present, to just click on the Create New, and you are able to create the chemical. And finally, it's the same deal for the signal. This process is pretty straightforward. Finally, you just click on Submit Metadata, and now it's going to upload to Flapjack. Is there any questions for the moment while we wait for the uploading the file? Okay, I guess they are not. Just to let you know, this process, you're going to do it again on the pod with Guillermo in order to explore the database once you have created your local installation of Flapjack. Okay. Now your study has been uploaded to Flapjack. We can now see all the managed options that you have for your studies. We are now in the Browse section, as you can see. If we are the owners of this study, and only if we are the owner of the study, we will see the Manage button. Here we can click. The first option is Share. With this option, we can share the study with someone that has already created an account on Flapjack. We just need to state the name. For example, here I can share this study with Guillermo. And it's easy as that. So now Guillermo has access to the study to manage the study and make analysis as they wish. You can also make the study public. So now everyone can see this study. Keep in mind that these two options are independent. So if at any given point, I change my mind and I don't want this to be public anymore and I want to make it private, Guillermo is not going to lose access to this study. And finally, we have the option to delete. You have to be careful with this option because there's no going back from deleting a study. Finally, we can go and view in a study. For that, just go to the View section. Here, we have to create a new tab. How are you going? Are we good for the time? Do you need more time? Okay. Well, first we create a tab and we can rename it. For instance, in this tutorial, we're going to review context effects. So I'm going to change it to context effects. And then we select the study that we want to analyze. In this case is context effects. As you can see, we have now the autocomplete feature. So you don't have to click every single assay and vector. It is automatically done for you. We're going to use all the assays for this, but for the vectors, we're just going to view the PAAA vector. So I'm going to unselect all the other ones. On the strain, I'm going to view MG1655Z1. The media is M9 glucose. And since we're going to use just to view the raw data, we're not going to perform any analysis. On the plot options, we're going to normalize by minimum/maximum. The subplots are going to be the vectors. All lines are going to be signal. And the plot is going to be the mean. Yeah, now we click plot. And we can see the raw data for this study. Now, if we want to make an analysis, we just need to go to the analysis section and select the type of analysis that we want to perform. For instance, in this case, we're going to select expression rate direct. We're going to give the default values, and the biomass signal is going to be the optical density. We click plot. And now Flapjack is processing to make the analysis we asked. So now you can see the plot with the analysis performed. You can see that this plot is interactive. So you can see the values when you're moving your cursor at each given point. You can also zoom in to see the data and auto scale it and do a bunch of different stuff with the plot as you please. You can also download this plot by just clicking the download button. You can select the screen format. You can select paper format. In this case, it's going to download it as a JSON. And you can also download it as a PNG. A PNG is intended for you for a publication, for instance. So it's easier for you to specify the font size, the width, the height, and the line width that you want the plot to be in. You just click download, and you will be able to get the plot in a PNG file. Another analysis that we can perform is a gamma graph. And for that, I'm going to add another tab. As I do previously, I'm going to rename it. Oh, sorry, I made a spelling mistake. In the study here, we're going to use TCAS9 inverters. We're going to use all the assays. The vector, we're just going to see the effect on 1. Then we have to go to the analysis section. And here we select graph at the very end. The chemical in this case is IPTG. and the function is expression rate direct. Now we have to specify the different values. For this case, the reporter recordation rate is going to be 0. The significant value is going to be 1 to the minus 6. The standard deviation is going to be 2. The minimal biomass is going to be 0.05. And the biomass signal is the optical density. For the plot options, we normalize by none. Our signal is going to be vector. The lines are going to be, subplot is going to be signal, I mean. The lines are going to be vector, and we're going to plot the mean plus minus the standard. And now Flapjack is going to perform the analysis for you. How did you find the interface so far? Are you having any troubles? Did you find it intuitive to use? Any feedback is appreciated. And now you can see the camera graph here. And all the previous tools are available here. So you can zoom, you can interact with the graph, seeing the values as you are moving your cursor. And of course, you can download it. And I think that's all. Now you know how to use the Flapjack interface. Any questions? There's one in the chat. Okay, first, can you add data about any custom signals / measurements as well? Okay. Maybe Tim, you can help me with that part.
Yes, you can. So you can add a description of the signal. So for example, you could put the wavelengths of the filters in the signal. You can also do kind of aesthetic things like choose the color of the signal. So when it's plotted, for example, GFP will be green. So at the moment it's left fairly open, that you have a description where you can specify information that you like. Because different types of signals will have different types of information that you want to add.
So I guess along those lines, so you mentioned you can add, like, let's say the wavelengths and even the filters, for instance, all of that is possible too?
Yeah, well, it's just an open description text field, okay, essentially. So you can put whatever you like.
Oh, I see. Okay, so it's not like a customizable key value pair. Is that correct?
It's not like specific fields for wavelengths that you could search, for example. But you could search the description, but it's just an open kind of text description, so it wouldn't necessarily match exactly what you're searching for. But in principle, you could search a description which has, a particular wavelength in the description.
All right, thanks.
And Jet, I think we should have somewhere in the GitHub repositories example Excel files that you can take as a template and can just modify them, remove the data tab, for example, and put your data in there. And by the way, just to mention that this is just one type of data, is the plague reader data, but as Gonzalo showed, there are other ways to upload data, and Gonzalo is going to show later via the Python package interface to the API or directly through the API. You can upload any kind of data that you like.
And I think that goes directly with the questions that on a related note, can you do bulk uploads too? I think that it's easier to do using the API.
Yeah, so at the moment through the front end, there's no bulk upload. The difficulty with bulk upload is that each assay, so for example, I might have 10 assays that I want to upload in bulk, but you'd have to describe the metadata for each assay individually, so it's a bit tricky. But we could think about that in future. It would definitely be a useful feature. For example, if you have assays which have common metadata, you could say, you know, all my 10 assays have the same layout, etc. And here are all the files for the data. It's definitely easier at the moment to do it via the API and the Python package.
Okay, if there are no more questions, the stage is yours, Gonzalo.